Here we are with lab presentation 8, and we're beginning then on page 404 with figure 52.3. Notice that everything on this figure and everything on the figure down below has been labeled for you. And notice also that 52.3 is a later stage of development compared to 52.2 on the previous page. You can see then that the figure is labeled in terms of a title as being a human embryo with extraembryonic membranes. And let's go ahead and start there. So take a minute and find those 3 extraembryonic membranes that we saw in the previous figure in 52.2. And so now you're looking for the yolk sac and the chorion and the amnion. And then take a minute and find the 4th extraembryonic membrane. And this then is the structure called the allantois. You can see the label kind of top center of the figure. And again, you might just want to jot down then as a reminder that the allantois is the 4th extraembryonic membrane, along with the yolk sac and chorion and amnion. The allantois, again, is the 4th extraembryonic membrane. Take a minute now and locate the fluid-filled amniotic cavity. And you can see then this cavity is surrounded by, is enclosed by the amnion. And notice then that this amniotic cavity has grown relative to the surrounding extraembryonic cavity. So if you compare this figure with the previous figure with 52.2, notice again now that the amniotic cavity has become much more prominent, relatively much larger. And again, at the same time then, the extraembryonic cavity is basically disappearing. Next, take a look at the chorion, and as we did in the previous figure, go ahead and trace this chorion now, this surrounding structure, and trace it over to the right-hand part of the diagram. And you can see then that here, the chorion, this expanded portion of the chorion, becomes part of the developing placenta, which is labeled for you down in the lower right. And recall now, these finger-like projections that we're seeing that are making up part of the developing placenta, these are those chorionic villi, which we talked about in the previous figure. And again, you can see they've grown much larger. You can see also they're not labeled for you here. But again, make sure you're good with the finger-like chorionic villi. And then notice in the very lower right-hand part of the figure, the endometrium is labeled. And of course, the endometrium, as you know by now, is just the deep part, the inner lining of the uterus. And the endometrium then, again, it's labeled lower right, and it extends up basically along the entire right-hand portion of the figure. Notice also then, and we're still on figure 52.3, we have these so-called maternal blood vessels. And they're labeled for you in the upper right-hand part of the figure, but then notice as you continue down the middle part of the figure, we have repeats, and so we have this series of maternal blood vessels. And these then will be carrying blood to and from the villi, the maternal blood vessels again. And you may recall this shown really, really nicely in the video, in the Life's Greatest Miracle video. You may recall at one portion there was a great shot of the villi, and you could see maternal blood essentially washing over the surface of the villi. And the narrator mentioned, and you don't need to put it down into your notes right now, it's more of a functional thing, but the narrator mentioned that as this maternal blood is washing over the villi, they were, or they would have been, the villi would have been, I think he used the term grabbing oxygen from the maternal blood, and they would also be grabbing various nutrients. But again, in terms of tracing blood flow, then we have these red vessels, these red maternal blood vessels leading to the area of the villi. And you might just want to jot down here in your notes that each of these red maternal blood vessels, and of course the red implies carrying oxygenated blood, so each of these red maternal blood vessels that we're seeing is referred to as a maternal arteriole. Again, then take just a minute and just label a couple of these red maternal blood vessels as maternal arterioles. And again, now picture that maternal blood, that mother's blood, washing over the villi. So now let's go ahead and talk about what's happening with oxygen that the mother's blood is carrying, and what's happening with nutrients that the mother's blood is carrying. So now what I'd like you to do is locate again just one or two or three of the chorionic villi. And now then I'd like you to picture the oxygen and the nutrients in the mother's bloodstream moving across the barrier, moving across the membrane surrounding the villi. And again then picture the oxygen, picture the nutrients moving into the blood vessels, which you can see within the villi. And notice then some of these blood vessels within the villi are red, indicating that they're carrying oxygen. And if you trace these red vessels along, you can see that they lead to the umbilical cord. What I'd like you to next picture then, continuing with these vessels carrying oxygen, carrying nutrients, again, they're going to be supplying the developing embryo with oxygen and with nutrients. And at the same time, waste products, carbon dioxide, for example, other waste products are going to be flowing away from the embryo. And so now, again, find the umbilical cord, find the large blue vessel leading away from the embryo and through the umbilical cord. And that large blue vessel, again, the blue implying it's carrying deoxygenated blood, blood low in oxygen, blood high in CO2 and other waste products. And again, now notice that that blue vessel, as it makes its way toward the villi, gives off smaller and smaller and smaller branches. And now imagine the oxygen and the other waste products leaving the blood vessels in the villi, and picture those materials making their way across the membrane that encloses the villi, and picture these waste products now dumping back into those pools of maternal blood. And then finally, to complete this blood flow process, notice from each of these structures that contain pooled maternal blood, and we're seeing, let's go ahead and count then. So look right where it says maternal blood vessels, upper right, and then just to the left of that, notice there's a chamber, and so this is one of those areas where blood is pooling. And then notice a little bit further down below, there's a second chamber, a second space, and then a third one, and then a fourth one, and then a fifth one. And again, now picture carbon dioxide, picture other waste products leaving the villi, making their way across the membrane surrounding the villi. These materials now again moving into these spaces that contain pooled maternal blood. And then finally, notice the small blood vessels, the small blue blood vessels, leading away from these spaces. And picture now then that oxygen poor, waste product high blood going back into the mother's circulatory system, into the mother's small blood vessels, and imagine then those small maternal blood vessels merging off to the right somewhere, far off, far away from what we're seeing in the figure. And imagine these smaller vessels merging to form progressively larger and larger and larger veins. And of course, now the blood is going to be back on its way to the inferior vena cava. This doesn't all have to go into your notes, but again, picture it, this merging of these very small blood vessels into progressively larger and larger and larger vessels, and finally the blood then going into the vena cava, going back to the heart, and completing a pulmonary circuit that you guys are familiar with from A&P 1. And keep in mind here that while the mother's blood and the embryo's blood are very close to one another within the placenta, the two bloodstreams never actually mix. And so while materials are being traded back and forth, oxygen and other materials, while these are being traded back and forth between the maternal blood pooled in those spaces and the villi on the other side of the membrane, separating the spaces from the villi, the two bloodstreams never actually mix. They trade stuff, but they never mingle. And we'll stop here and start presentation 9, lab presentation 9, with figure 52.4.